Hello, this lecture is on section 2 of your science resource, which is about, in this case, it's genetics. So the last section, there's only 3 sections in science. Section 1, we talked about cells, who discovered cells, and then we talked about all the parts of a cell, and the cell cycle, the life of a cell, whatever. This section is about genetics. So how is stuff getting passed down, what's getting passed down, how do you calculate what's being passed down? So when we talk about genetics, obviously we're talking about genes, and we're talking about stuff that you should have read in your material already, and this is kind of like a summary of it. Obviously, again, in these PowerPoint lectures, I can't cover everything. It's very important that if you want to succeed in the decathlon that you read through that section. So this is going to be like a good refresher for you. So first of all, the section in the resource guide, they talk about the basic definitions that you need to know to understand genetics. So I'm going to go over some of those definitions now. First of all, a somatic cell, somatic meaning a body cell, or basically any cell that's not a sex cell, a somatic cell has pairs of chromosomes. We talked about chromosomes, and these pairs of chromosomes, they get one from each parent, one from the mom, one from the dad, and those are called homologous. Homo, the prefix meaning the same, so the same kind of chromosome, one from the mom and one from the dad. Each cell has 2 copies of the same gene, so again, each one has the same. Each one has a chromosome from each parent, and in that chromosome, you also have the same gene, one from each parent. These same genes, or the pairs of genes, the one you get from your mom and your dad, those are called alleles. Okay, and then the 2 alleles, the one from the mom and your dad, depending on which alleles you get, will tell you which traits you're going to express, and that's called the genotype. So, and if the 2 are the same, the genotype is considered homozygous. Homo, again, the prefix mean the same. So if the 2 alleles are the same, like your mom has brown eyes, your dad has brown eyes, you express brown eyes, then that means your genotype is homozygous. But if they are different, like if you get a brown and a blue eye, but you express brown, but you have 2 different colors in your genes, that's called heterozygous. Hetero, the prefix meaning different. So here is an example just to show you this little X thing right here is a chromosome. Wound up, what makes up this X is basically a lot of wound up, like think about yarn, like totally tied together. That whole chain is DNA, and pieces of DNA, like if DNA was a wall, the bricks of the wall are called genes. So who first was, I guess, considered the father of genetics is called Gregor Mendel. He studied a pea plant. The actual scientific name that you need to know is Pisum sativum, a pea plant, and he used this it says plant hybridization experiment, so he crossbred them. He studied 7 traits. Obviously, you need to know all 7 for the decathlon. You could go seed shape and seed color, pod shape and pod color, so that's kind of together. Flower color, and they don't have flower shape, but they have flower position. So color shape, color position, and then the plant height. You should need to know all 7 traits that Mendel studied. Now, he studied, basically, if you look at this table, this is also in your resource guide, this is what he noticed, that each trait for a plant had only 2 different possibilities. For example, I'm not going to go over all these, but for example, the flower color could be either white or it's violet. There was nothing really in between. The pod, it could either be full or it could be constricted. There was no in between. So that's why he chose the pea plant. It was actually a very smart and strategic choice by Mendel to choose pea plants, or the Pisum sativum. So his scientific question, when he was doing the scientific method, and was trying to figure out how are traits transmitted to offspring. But in actuality, what he was really doing was trying to find or make a new crop that was profitable. He wanted to make money off of this. So like scientifically, we're like, oh yeah, he wants to know how smart or how he wants to know how smart or prove how smart he is, and figure out how traits are transmitted. But practically, he was trying to see if he could do this correctly, how can he just make the right crop like 100% of the time. So first I'm going to have you watch this quick 4-minute video. We're going to talk about Punnett squares, what he did. But before we talk about that, I want you to get a good intro to Punnett squares. I'm going to watch a quick video, again, what a Punnett square is.
Hi, my name is Mary Poffenroth. I'm an adjunct professor of biology, and today we're going to have a little fun with Mendelian Punnett squares. Now, before we get started and jumping into our Punnett square problems, let's set down some ground rules. First, we're going to talk about alleles. Alleles are going to be like different flavors of genes. So, go into the ice cream shop. There's 31 flavors of ice cream. even though they're all different flavors, they're all still ice cream, right? Alleles are going to be those different flavors of genes that you have available to you. The next thing I want to talk about is dominant and recessive. In any relationship, some of those alleles are going to be dominant and some of those alleles are going to be recessive. Those dominant alleles, if they're present, they're going to have more of a say in what the outcome of that class is going to be. And the recessives, well, they're going to have little to no say unless there are two recessives that are available. Also, in each of our crosses, we're going to have one allele from our mother and one allele from our father. Now, let's jump into our crosses. Today, we're going to do a 4-square Punnett square. So, you want to start out by just drawing a square. In that square, you're going to draw a line right down the middle from the top to the bottom and a line from left to right. Now, you have 4 individual squares. Each of those squares is going to be a probability, and each of those squares is 1/4, right? Or 25%. Next, we're going to put our parents' alleles on the Punnett square. Now, it doesn't really matter if you put male or female on the top, but generally people put male on top, female to the left. For our problem today, we're going to use eye color. The eye color alleles are going to be, for our father, it's going to be brown phenotypically expressed alleles. That means that father has brown eyes, but his genotype, or the genes at play, his alleles are going to be big B, little b. That means that he's heterozygous, or he has 2 different alleles for that trait of eye color. It's a little important to note at this time that all of these letters are completely arbitrary. You can make up any letters you want. However, in any relationship, a capital letter or a big letter, like say big B, is going to be for the dominant. And a little letter, or a lowercase letter, in this case little b, is going to be for the recessive. Okay, so we have the father, big B, little b. He's heterozygous, meaning he has 2 different alleles. What about the mom? The mom has blue eyes. So phenotypically, her expression that you see on the outside is blue eyes. Her genotype, or the genes at play, are going to be little b, or homozygous recessive. Alrighty, it's time to do our cross. Now, we're going to take one from the top and one from the left, and we're going to bring them down into those squares. So, we have on our top, big B, little b, then bring another big B, little b down. In our right square at the top, we have little b, and our bottom right square, we have little b. Okay, what does that mean for the probability of getting an offspring with what kind of eye color? Now remember, each of those squares is going to be 25%. So, we have a 25 plus 25 or 50% chance of getting an offspring with brown eyes. And we have a 25 plus 25 or 50% chance of getting an offspring with blue eyes. And for a fun little extra problem when you're sitting around the dinner table, take the eye colors or the phenotypes of your parents. Even though you won't know the exact genotypes or the genetic makeup behind what you're seeing on the outside, do some crosses and see if you could find out the probability that you could have actually had a different eye color. Thanks for watching, and if you want to learn more about this subject, click on the link.
So those are the basics of Punnett squares, and if you didn't get that, I showed the video because it's very, 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 very basic. I'm gonna go over it again and what Mendel actually did with those pea plants. So first thing he did was he thought to himself, let me breed a pure purple plant and a pure white plant. So if I do that Punnett square, if I look at the top row, I have a pure purple, capital P, capital P. And on the left, I see little p, this is pure white. And then I cross them, just like they showed in the video, I get big P, little p in all 4 boxes. Which means, so here's the homozygous dominant, homozygous mean the same, and dominant. Here I have homozygous recessive. And then the kids, they call this the F1 generation. So the first set of kids, they call it F1. And they notice that in the F1, 100% of all the kids will have a heterozygous genotype. Capital P, little p, 100%. So even though there's a purple and the white, even though mom and dad, purple and white, they all end up being purple. So the next thing they said was, what if we mixed, and then the phenotype, the term phenotype we haven't talked about yet. Phenotype is what is actually being expressed. So genotype is capital P, little p. Phenotype is purple. Okay, so phenotype is what you actually see. Genotype is what you actually have. What alleles do you have? So he said to himself, now, what if I cross the offspring of the F1 generation? So if you remember, the F1 generation was heterozygous and heterozygous, capital P, little p. And if I cross, you notice I get capital P, capital P, lower p, capital P, lower p, and I have two lowercase p's. This is called the F2 generation. Now here's where it gets interesting. So this one is all purple, this one is all purple, but 25% will get pure purple, capital P. Another 25% will get lowercase p, which means white, which is again at that time very crazy to think that you have a purple dad, a purple mom, and all of a sudden you have a white kid. Obviously people think like, oh, there's a problem, or maybe albino, but really that's how the genes work. And 50% will be heterozygous, capital P and lowercase p. 75% phenotypically will be purple, these 1, 2, 3, and then the 25% phenotypically will be white. So next thing that he did was called a dihybrid cross. What if he compared two traits at the exact same time? So he says dominant is round and yellow and recessive is wrinkled and green. And just to save time, this is what he did. He crossed them all. A purebred dominant, capital R, Y, capital R, Y, and then this is a purebred recessive for the other parent. Then when they cross the F1 generation, this is what they get. What percentage would have both round and yellow seeds? You know, capital R and capital Y. They all do. So 100% of the F1 generation will have round and yellow seeds. Just as you noticed before, what if he cross-pollinates the F1 generation? So same Punnett square, but I have different genotypes. And when he crosses them, he says how many will have both round and yellow seeds? Well, I see here 1, 2, 3, 4, 5. This doesn't have a capital R and Y. This doesn't. 6, 7, 8, and 9. So 9 of the 16 possibilities will have the dominant gene. So again, crazy at that time to think that, again, if you have a purebred or you have this stuff and or you have a round and yellow seeds and you combine it with another round and yellow seeds and then you don't get round and yellow seeds in their kids. This is basically what genetics is all about. So here are the conclusions, and one of them is silly. I don't know why they did it. But first of all, the recessive trait, if you have a purebred and a purebred, the recessive trait disappears in the F1 generation. This is some silly one about alpacas. Alleles, they sort independently of each other. So you may notice that in the last example with the dihybrid, it doesn't matter if I combine two traits at the same time, like it still worked exactly the same. So it's basically alleles sort independently. And the last thing is that the results from the monohybrid cross, or everything that we just saw right now, had no bearing no matter what gender they were. They could be male or female or whatever, but they'd still get the exact same results. So it's gender independent. So here are the 3 laws that came out of these experiments. The first law is law of dominance, the second law of segregation, and the other one, the law of independent assortment. Law of dominance means that there's going to be one allele that dominates another. So when you do genetics, there'll always be one allele that dominates another. And that dominant allele determines the phenotype. So if the dominant allele is purple, it will show purple. The law of segregation says that before fertilization, and this is before he even knew about meiosis, that before fertilization the alleles get separated or segregated, and then when they're fertilized, when a man and a female come together, those alleles are brought together again. So this is saying that the alleles are separated. And the last one, independent assortment, they separate independently of each other. So basically from that dihybrid cross that we just saw, it's basically saying that the alleles separately or independently separate. They don't like combine together. They don't join together. Everything is all separate. All the genes, all these alleles, they all work separately. And those are the 3 laws from Gregor Mendel. So Gregor Mendel, the great things about that he did, first of all, we talked about it in the last lecture in class. At the time, people thought that there was going to be a blending inheritance. So if you have a purple dad and a white mom, maybe you'll have a purplish, whitish, or an in-between color. That's not true. Traits are passed down. Traits are not passed on to the next generation, but genes are. And then he noticed that with the F1 generation. So when he did pure purple and pure white and he got all purple, he basically rejected like, hey, there cannot be anything, there's no such thing as blending. The alleles, he talked about, again, we have two alleles, basically two versions of each gene, or flavors of each gene, one from the mom, one from the dad. And then dominance. Again, some alleles are dominant to others, and we'll talk about later that it's not always the case with genes, but for the most part, some alleles are dominant to others. The law of segregation, which we talked about, that they segregate during meiosis. And in hybridization, his dihybrid cross, remember the R's and Y's together, it led to the law of independent assortment. So he did this for a while, and it took like 34 years until his work got verified and actually accepted by the scientific community. Here are three names you need to know. Hugo de Vries, he came to the same conclusions as Mendel in 1900 and said Mendel was right. Carl Erik Correns, he also experimented with garden peas, and he proved the law of segregation and independent assortment in his paper in 1900. And then Erich von Schirmack-Senseneg, he also confirmed that 3-to-1 ratio. So if you remember the F2 generation, how you had 3 purple and 1 white, he also confirmed that ratio in his work. So 30, 40 years later, these three scientists, in fact, validate everything that Mendel said. So that's everything about Mendelian genetics. That's everything you should know about that. This next section, they talk about in your resource guide about genetic diseases and stuff like that. Basically everything we talked about, but applied to what we know about us humans. So the first thing, autosomal recessive genetic diseases. Autosomal, you remember there's 23 pairs of chromosomes, and one pair of chromosomes actually determines your gender. All the other chromosomes, the other 22 pairs that don't have to deal with the sex, they call that the autosomes. Okay. So here are a couple that we're going to talk, a few diseases that you need to know. And they talk about this in your resource guide. First one is called alkaptonuria. It was the first genetic disease discovered, okay, alkaptonuria. And basically there's an enzyme that is defected because of a mutation. So alkaptonuria. Next one is cystic fibrosis. You may have heard this because it's a little bit more popular. Basically there's a channel protein that's defective, and you get a buildup of mucus and lungs in the lungs and the digestive system. So basically a protein is defective and it tends to build up mucus, or basically there's a log jam and you can get these big lumps in your body. Sickle cell anemia you may have heard of in your bio classes or may have heard before, but red blood cells in our body, they should look like red, soft, pillowy marshmallow donuts. But because there's a mutation, the hemoglobin folds incorrectly, and instead of looking like these soft jelly donuts, they look like sickles, sharp, like almost like blades. Because they look like blades and not donuts, they can't deliver oxygen as well as normal red blood cells. So that's called sickle cell anemia. And the last one, Tay-Sachs disease. You have a lipid metabolizing enzyme. So remember, lipids are like fats, and you have an enzyme that metabolizes them, that breaks them down. Unfortunately, it's disrupted or defective, and the lipids, instead of being metabolized, they surround neurons. Remember, neurons are cells in your brain, and brain signals are not being transported back and forth like they should. So that's Tay-Sachs disease. 4 diseases that you need to know. I think the three last ones are kind of easy to remember. The first one, alkaptonuria, is new to me also. But remember that's the very first genetic disease discovered. If you are heterozygous for any of these diseases, you're called a carrier. So even if it's recessive in your body, you are considered a carrier because, again, you could have another recessive mate and your son or daughter can contract it even though neither of the parents have it. So you're considered a carrier if you're heterozygous for any of these diseases. We talked about the law of dominance earlier. We talked about how in alleles there has to be one that's dominant, but there are exceptions to every rule. There are 2 exceptions to the law of dominance. One is that maybe the allele is neither dominant or recessive. Or there could be that both are dominant and neither is one like better than the other. If you have the case where no allele is dominant over the other, they call that codominance. And if neither allele is dominant or recessive, they call that incomplete dominance. So here, a great example we'll talk about that is blood type. Some of you may know your blood type or have given blood before. This is considered codominance, where if I have an A and an A, you you express capital A, capital A. You express both of them. That's called codominance. Incomplete dominance, meaning that neither is dominant or recessive. This is a good example of that blending theory that we talked about. So here's an example of these flowers of a red and a white. And if they got heterozygous for red and white, it ends up turning pink. Again, it's not very common, but this is called incomplete dominance, where it's not one or the other. There's actually a mix between the two. So one thing we're going to talk about is familial hypercholesterolemia. As it sounds like, it's talking about cholesterol, and this is one genetic condition that someone can have. This is the normal process in your body. Normally you eat food, like fatty foods, or maybe you eat fries, or eating Zavin's burritos, and you get a whole bunch of cholesterol in your bloodstream, and it's not good in your blood. So what happens is that it binds to a protein called LDL. So you have these proteins in your bloodstream called LDL, and cholesterol binds and sticks to it. After that, the LDL, that protein, binds to a receptor for that protein, and then the LDL complex is brought into the cell for delivery. So basically, the cholesterol binds to a protein, and this protein enters a cell, and the cholesterol is removed from your bloodstream. But if there's a mutation of the LDL receptor gene, then the pathway does not work correctly. If it doesn't work correctly, that means no cholesterol is going inside of a cell or binding, which means there's so much cholesterol in your bloodstream. And it determines, depending on what mutation that you have, it could be very severe. The disease can be very severe. This one is an example of an incompletely dominant genetic disease. Remember we talked about incomplete dominance? If you look back, incomplete dominance meaning that it is a mix of the two. So in this last bullet point, where it says the extent of the mutation determines the severity of disease. So if you have, it's not you have it or you don't. In this case, is if your mom has a little of it and your dad has a lot of it, you'll have a middle ground severity of this problem. So hypercholesterolemia is incomplete dominant genetic disease. It's a good example of incomplete dominance. An example of codominance, as I talked about earlier, is blood types. Okay, so this one was discovered by Carl Landsteiner, and this is the best example of codominance you'll ever hear about. So thinking about two antigens, A and B, so you may have heard of that, I'm blood type A, blood type B, but they're expressed at the exact same time. So here's a good table for you. If you're group A, or blood type A, this is your red blood cell type. You have basically circles around each blood cell. The antibodies in the plasma are anti-B or not B, and you have an A antigen on the outside. So see, this little pink lollipop matches the outside. If you're a group B or a B blood type, you have diamonds instead. You're anti-A and you have a B antigen. If you're blood type or you're group AB, you have both the diamonds and the circles. You have no antibodies and you have both of these antigens. And group O, you notice it looks kind of naked up here. It has anti-A and anti-B antigens, and there's no antibodies, and there's no antigens on the cell whatsoever. And then you may notice that if you ever got your blood type checked, you can get positive or negative. So me personally, my blood type is B positive. They call this the rhesus factor or the Rh factor. So there's another surface antigen on the red blood cells. So when we talk about surface antigens, we're talking about this stuff on top of the cells. So there's actually another one called the rhesus factor. It's named after this monkey. This one on the left, it's named after this monkey. So if anybody in the test asks you what's the rhesus factor named after, it's named after a monkey. And it follows simple dominant patterns. So just the normal, plus is dominant, minus is recessive. Why do we care about the Rh factor? Well, it can cause a serious issue if a recessive mother is pregnant with a dominant fetus. So what happens is that the blood types are incompatible and the fetus will not get the blood that it needs, or it's not compatible with the blood that the mom is giving to the fetus. The baby is born safely, but future pregnancies will require extra attention. So that's why this positive and negative stuff is actually very important. And then, I think this is one of the last ones we're talking about, pleiotropy. Pleiotropy is a gene that actually influences other genes that are unrelated to it. So this headline, the trendsetter, is actually very appropriate. So Gregor Mendel noticed that when the pea plants with white flowers, its seeds also had white coats. And to him, it was kind of weird. Remember, things were supposed to be independent. How come every time they had a white flower, the seeds also end up being white? This is an example of pleiotropy. So another example is albinism. I'm sure many of you have heard of albinos, but this is a brown bunny, or as the bullet point says, this cute bunny can produce a brown pigment called melanin. Albino bunnies, however, have a mutated melanin gene. Because that gene is mutated, everywhere in the body is white. Or again, think of this as a trendsetter. This one gene, because it's mutated, affects all the other genes. It's not just one gene acting independently. So here's a quick video of some guy, I don't know where, I guess this is one educational video of him talking about pleiotropy. Oops. There we go.
And I want to talk to you now, about pleiotropy. Pleiotropy. What is pleiotropy? Pleiotropy is what I just described, when the expression of one gene has multiple phenotypic effects. And sadly, our best results of this, or excuse me, our best examples of this are in human genetic disorders. Multiple phenotypic effects. And the one I want to use is a human genetic disorder called sickle cell anemia. Sickle cell anemia is a very powerful one to use because it has an unbelievable number of sad effects to those that are completely affected by it. Quick review of sickle cell anemia. Sickle cell anemia runs in the disease is in 1 in 400 people of African American descent. And what it is caused by is a defective hemoglobin gene. Now the hemoglobin gene is, let's say it's big H. That would be a normal hemoglobin. So if you have a normal hemoglobin gene, you're going to express normal hemoglobin, and even if you are recessive, but hybrid or heterozygote, you're going to be phenotypically normal. However, someone who is little h, does not make any normal hemoglobin, and in making no normal hemoglobin, ends up with a changed hemoglobin molecule, and that hemoglobin molecule causes the entire red blood cell, because remember, hemoglobin is a protein in red blood cells. It causes that entire hemoglobin to twist or sickle. And that is the primary effect of the genotype, a defective hemoglobin protein. But it's pleiotropic. Because you see, there are multiple, what you and I call, in terms of human medicine, symptoms of this disease, but as geneticists, they are pleiotropic effects. What are some of those pleiotropic effects?
So that's the quick commercial version of a pleiotropy. I know I didn't really talk about yet why is it pleiotropic, but long story short is that just because that one sickle cell is, or that one red blood cell is changed to a sickle, it changes everything else. So think trendsetter, think albinism, where one gene changes the way other genes are expressed even though they're unrelated. And then they call this the star phenotype. It's called polygenic inheritance. You know the prefix poly meaning many. This is kind of the reverse in terms of one gene affecting all the other genes. This is basically many genes influencing one phenotype. So we've only talked about like an allele, one for the mom, one for the dad. But we could talk about many, more than two, affecting what is being expressed. So the best examples of this are skin color. So it's not, skin color is not just, you know, black and white and then you do the Punnett square. There are way more than two that affect what your skin color is going to be. Same thing with your weight. You should know that some families that are skinny tend to have skinny children. Some kids that are chubby tend to have chubby children. Many genes go into that, not just one gene. Same thing with height. And also even breast cancer as well. It's not just one recessive gene. It's more than one gene that determine whether you express it or not. And the last thing we're going to talk about is sex-linked traits. So things that are passed down. If you remember, we talked about autosomes. Remember, there's 23 pairs of chromosomes. 22 pairs are called autosomes, and the last pair are called your sex chromosomes, whether you're a male or a female. So one example is color blindness. Here's a color wheel. See if you could see if you're colorblind or not. If you're colorblind, you won't be able to see all these colors. But colorblindness is actually an X-linked trait. So males actually have a better chance of contracting colorblindness because they only get one X, whereas females, they get two X's. So say, for example, there's a woman who's a carrier for colorblind, and she's having a baby with a colorblind male. What are the chances that the daughter will be colorblind? What about a son? Well, this is how the Punnett score will look like. Here's the mom. Again, she's a carrier. Carrier meaning heterozygous. So let's say N is not colorblind and C is colorblind. So let's just say on her genotype, she has one N on an X and another C on an X. Let's say the male has color blindness on X, and remember, color blindness is not on the Y chromosome. Well, I cross, I get NC, NY, CC, and CY. So ultimately, the daughter and son, whether the child is a daughter or a son, they have a 50-50 chance of being colorblind. So for example, here I get X and X, and here I get X and X. So this is going to be a girl. This girl has a 50-50 chance. This one has NC and CC. And this one over here, XY and XY, we have the colorblind one here, meaning that it only has a 50-50 chance also of being colorblind. But for the most part, what you should know as well is that colorblindness is more frequently found in males because males only have one X. So if they get colorblind, they're going to get it. But if, like, for example, in this case right here, a female could have an N and a C, which means the girl would not be colorblind because she has two X's or two chances to not get it, whereas a guy only has one chance to get colorblindness. So here are the 46 chromosomes we talked about, all 23 pairs. So these first 22 are called the autosomes, and the last pair, the 23rd one, those are called the sex chromosomes. Okay, so females have two X's, males have XY, which is right here, and the Y chromosome contains something called the sex reversal gene, or SRY. Sex reversal gene, SRY. So sex reversal, then the Y gene. When this Y chromosome is present, then the fetus begins to develop all the male parts. Now, some of you may be curious to know, when you have kids, how can you figure out this on your own? Well, you can get genetic testing. First, there's two different types of analyses. One is called a pedigree analysis. It's not very invasive. It's the equivalent of looking at your family tree. So what you do is you look at your family tree, you see what your dad has, your mom has, their brothers, their parents, and it predicts the risk of passing on disease. It also identifies who in your family are carriers, and it's best applied to disorders caused by a single gene. But if there's a disorder by multiple genes, there's no way you can do anything with a pedigree. Again, a pedigree is basically a fancy word for a family tree. The way you could do this to be even more, how can I say this, more specific or more exact in what you're going to contract is called chromosomal analysis. It's while the baby is still in the womb and you take a sample of the fetus, which is called a karyotype, a fetal cell sample. So chromosomal analysis looks like this, and there's two different kinds. One's called amniocentesis, and in amniocentesis you take the fetal cells from the membrane. Amnio, you may have heard of like the amnio fluid. They take the fetal cells from the fetal membrane. The other one you could do is called chorionic villus sampling, and this one you take the cells taken from the fetal membrane, for a biopsy. So they take it and then they do a biopsy on it. So these are the two types of chromosomal analysis: amniocentesis and chorionic villus sampling. And that's it for section 2 for science. So we're 2/3 of the way done with science. Those of you that took AP Bio with me and those of you that took biology already, it should be a lot of review, and hopefully you don't find this too difficult. Again, if you have any questions, feel free to ask me, and I'll see y'all later.